AI and quantum computing is helping us solve problems like mapping the brain that would have taken us millennia before. Let's see what Google Research has to say on the topic.
So many thousands and thousands of neurobiologists, we still have a dim view, actually, of how brains work.
Ultimately, we're trying to understand one of the most fascinating and complex object we have in the known universe. I can't imagine anything more exciting than really finally figuring out how this little object that generates everything we experience actually works.
It's kind of cool that we're using the mind to figure out how the mind works, the essence of reality.
Connectomics is a word that we coined by accident. It was a play on the word genomic, which was a map of all the genes. We said maybe we could have a map of all the connections between nerve cells.
I'm a vocabulary nerd, and making a word like connectomics is very interesting to me because words are how we describe life, and sometimes other languages have better words to describe life than we do.
Understanding the brain is clearly a biological problem, but acquiring the map of the brain at scale is really a problem in imaging, computer science, and software development.
Even though the human brain may seem like a small object, if you start looking at it nanometer by nanometer, you end up with literally an exabyte of data, which is 1,000 petabytes, which is 1,000,000 terabytes, and so on. If we were to map the whole human brain right now, it might take billions of dollars and hundreds of years.
Which isn't feasible, especially under our current economic conditions.
So we know we need to start with understanding the brains of simpler things that we have a chance of actually figuring out in the next decade or two.
The first connectome was done on a worm known as C. elegans, and it was chosen because it has so few neurons, only 300.
We have to start with smaller problems, like babies do, in order to make the bigger problems seem more feasible.
The process for mapping neurons in C. elegans was to take very high resolution pictures of slices of the worm using electron microscopes, and then literally print out those pictures onto large micrographs and then annotate them by hand. So they would take a marker and trace the structure of one wire from one micrograph to another until they traced the thing across the whole worm's nervous system and did that for every wire. In some sense, what they learned from the worm is that this whole business is a pain. It took them 10 years to do a tiny worm with 302 neurons, so people kind of gave up pursuing additional connectomes for almost 20 years.
Definitely a very inefficient and slow process. I can't believe they even worked on that for 10 years. But it helped spur creation of this, so maybe it's not all for nothing.
It was not until the 2000s when massive technological upgrades to the process could make it much more feasible.
So we picked the fruit fly brain for this because fruit flies have interesting behavior. They're complicated, they have learning and memory, but their brain is only 100,000 cells.
300 to 100,000 is still a massive leap.
As researchers started to map the fly brain, they were generating massive data sets that involved a number of different computational steps to get from the raw data to actual knowledge that biologists could use.
I think it's fair to say that without Google Research help, we would have failed. They had the expertise and the computer power to process this amount of data.
These massive companies, although they have some downsides, there's definitely a bunch of positive upsides as well. They help us push innovation forward.
Without the technology that we and our collaborators have introduced, it would have taken millions of hours of human effort to finish even the fly brain. The main problem that we started working on at first was what's called the segmentation or tracing problem, which is where you have a three-dimensional data set containing all the wires in the brain, but you don't yet know which wire is which and where they go. We developed a 3D convolutional network approach, which was designed to vastly improve the process of automated tracing of 3D neurons.
Taking 2D, like how we see screens or how we write on paper, and making them 3D, like how we experience real life, is definitely progress in my mind.
Flood filling network, which is a coloring algorithm that basically colors in an object and then fills that object as if it's a volume. It's like pouring paint into a pipe.
In addition to that, we developed a visualization software called NeuroGlancer. People could finally view these data sets and their 3D reconstructions within the convenience of a web browser.
Taking that 3D image and being able to view it on a 2D screen is quite the concept.
One of the main things that we've learned from mapping, in particular the fly brain, is that structure seems to be very closely related to function. The shape of the neurons and who they talk to gives us huge clues as to actually what the brain is doing and what the purpose of those neurons are. This wasn't obvious before we started this research.
It seems like in all of life, structure implies function. We make things like satellites to reflect our ears, or cameras to reflect our eyes. Everything that we do, all the technology that we make, is reflecting the human body. And that's the same thing that's going on here.
So what we're really trying to do now is scale up. The logical next target for connectomics is a whole mouse brain. It's 1,000 times larger than a fly brain, but still 1,000 times smaller than a human brain.
Worm to fly, fly to mouse, and maybe mouse to human.
When you look at mouse brain in the electron microscope, it looks exactly like human brain. It is, in fact, a miniature version of a human brain. To do a whole mouse brain is not going to be trivial, unfortunately. It's going to require a ramping up of technology, both at the imaging side and at the computer infrastructure side. But it's possible that if we do a whole mouse brain, we will learn so much that we won't have to do a whole human brain because we will have figured out the essence of how information is stored in brains.
Now, this is where I disagree. I don't believe that we can find out everything from the mouse brain. I believe that eventually we're going to have to try to map the physical human brain.
What gets me excited is that one day we will have a precise understanding of how do we form memories, how do we recognize mental disorders or diseases. But in order to do that, we have to create this loop of technology that's, a little bit unimaginable even to people a few decades ago.
The steps seem to be moving faster and faster, which makes the future a bright one. If you liked listening to me yap, hit those like and subscribe buttons, and I'll see you in the next video.